Okay, let's start the class. Okay, so last class what we discussed korisilu, we have talked about, or we have started the cell division called as meiosis, and what we remember, meiosis is also called as a reduction division, right? It completes itself in two steps, that is meiosis 1 and meiosis 2, and we have completed prophase 1 of meiosis 1, right? So prophase 1 boli kole, again prophase 1 has been divided into certain stages. Ki stage asile? Keta stage asile prophase 1 or pasta. What are the 5 stages? Leptotin, zygotin, bacchitin, diplotin and dikinases, right? And we remember the important characteristics that occurs in each stage. Whenever we talk about leptotin, the first word that we remember is leptos mane ki, thin threads like structure, no? So the genetic material always appear in the form of a thin thread, right? And then we have certain words called as the bouquet stage, beaded appearance, etc., etc. Hekhini asile leptotin. Talking about the next stage, that is zygotin. What are the important points? Bivalent synapses, synaptonymal complex, right? As far as bivalents are concerned, we need to remember one line, no? The number of bivalent in a cell is equal to, it is equal to the haploid number of chromosomes of that cell, right? So twice n jedi 46 hoy, n jedi 23 hoy, tetele bivalent kiman hobo 23, right? Next, after zygotin, we have talked about the next stage, that is bacchitin. So what's the important characteristic of bacchitin ki? They crossing over. What happens in crossing over? Exchange of genetic material. We have talked about something called as the recombination nodules, right? So recombination nodules ki asile, it is the site where the crossing over will take place, right? After the exchange has been done, then what happens is the chromosomes will tend to separate, right? Sister chromosomes, non-sister chromosomes, sister chromatis, non-sister chromatis, these are the words that we have discussed, right? After exchange, then they will tend to separate. Talking about separation, instantly separate hobo, ne gradually separate hobo. Gradually separate hobo, we have talked about the next stage, that is ki bacchitinor pisot diplotin. Diplotinot ki hoy? Chiasmata formation, that X-shaped structure, and finally we have the last stage, that is dikinases. What happens in dikinases? Terminalization, and plus it is this phase where the nuclear membrane, nucleolus, etc. will disappear gradually, right? So once prophase is over, what happens to the chromosomes? We see that the chromosomes remain scattered in the cell, because before to this chromosomes were within the nuclear membrane, but after the nuclear membrane disappeared, chromosomes will scattered in the cell, and after prophase, as we have already discussed in case of mitosis, right? Prophase of the next stage is obviously metaphase. So similar is the explanation in case of meiosis also. So what will happen in metaphase? Junbilak scattered chromosomes asle gutebur ahi pelai equatorial planet arrange hoye jabo, right? So aje likha metaphase. Metaphase likhile hobo jano? Metaphase 1, exactly. So metaphase ki hobo bivalents bilak equatorial plane arrange hobo. And what is the number of bivalent that we are drawing in the diagram? Only one pair, right? Not necessarily okol eta pair e hobo. Actually hoy ki, so if you are talking about this one, tete hala aru eta pair thakibo, akko eta pair thakibo. Cell to jiman bivalent asile, suppose I am talking about a cell that has 3 pairs of bivalent. Yate ami okol eta dekhaisu de, it's just a diagrammatic case, and so we are just showing you only one bivalent just to understand. Is the point understood? Right? Actually ki hobo jiman asile no, 1 pair, 2 pair, 3 pairs, right? Depending upon the number of bivalent that are present in a cell, all the bivalent will remain they will get arranged in the equatorial plane. In the diagram what we are showing is just one pair of bivalent, right? Now don't get confused by this diagram, say okol cell to eta bivalent ase niki, nohoy tenuka bahut bivalent ase, but the same diagram. What is the point of drawing the same diagram again and again, right? So we know exchange has taken place. That is the reason why we are talking about two colors, right? So exchange hoy gol, exchange hoy jawar pisot bivalents bilak ki hobo, arrange hoy jabo. And what is this? Nothing but the spindle fibers. Spindle fibers form koribo. Spindle fibers, I think you remember we have talked about the centriole, the astral rays, the continuous fiber, the discontinuous fiber, etc., etc., right? So if you are talking about this, etu ki hoy discontinuous fiber, কারণ ইয়াতে ভাঙি গৈছে আৰু এইবিলাক কি হয় কন্টিনিউয়াস ফাইবার্স নেক্সট লেখা Anaphase 1. Okay, we have talked about anaphase during the process of mitosis, and what we remember, what happens in anaphase? Anaphase or ki hoy? Mitosis or anaphase we have already studied, no? Meiosis, anaphase 1 we did not study, that's okay. But mitosis we have talked about anaphase, anaphase or ki hoy? Just in one word, if you want to describe the phase, how will you describe? Anaphase or ki hoy? Separation, right? Or else you can be using a word is splitting of the centromere. Right? So these are the things that occurs in anaphase, but the point to be remembered in case of meiosis is the centromere does not split, but the bivalent splits, right? One bivalent will move from one pole, the other bivalent will, one of the bivalent or one among the two will move to one pole, the other to the two poles. In this case, the centromere will not split. What do you understand by centromere splitting? What will happen if a centromere splits? Eta chromosomor centromere to split korile ki hobo, chromatids bilak separate hobo, right? Yate in case of meiosis 1, the centromere splits or this does not split? It does not split. If it splitted, tetale etu part separate hoy jabo dokhon, right? So in this case, the centromere does not split. What splits is the bivalent, right? read the line whether it's understood or not. এটা bivalent প্ৰথমতে কি হৈছিল exchange হোৱা নাছিলে exchange of genetic একদম origin যেতিয়া we are talking about the first stage that is zygote sorry second stage that is zygote bivalence were formed but exchange did not take place exchange ক'ত হৈছে পেকাইটিনৰ পিছতহে হৈছে so পেকাইটিন onwards আমি exchange বা recombinance কিটা পাম নে নাপাম right আৰু সেই recombinance বিলাকত কি থাকিব both parents characteristics থাকিব ক্ৰোমোজোমৰ ৰেপ্লিকেশ্যন ক্ৰোমোজোমৰ সংখ্যা চোৱাচোন ইয়াতে কিমান আছিল দুটা আছিলে এটা দুটা মানে কি এটা এনেকে পেয়াৰ আছিলে ন ৰাইট এতিয়া কি হ'ব এনাফেজ এটাতো মেটাফেজ হৈ গ'ল এনাফেজত কি হ'ব এই হাফ ইয়ালৈ আহি যাব হাফ তালৈ যাব নাযায় তেতিয়াহ'লে কি হ'ব ক্ৰোমোজোমছ কি হ'ব কমিহে যাব ইয়াতে ডাবল আছিলে এতিয়া এটা ইয়াতে তিনটাহে গ'ল এইপিনে তিনটা আহিলে প্ৰথমতে কেইটা আছিলে ছটা তো ছটাৰ পৰা তিনটা হৈ যোৱা মানে কমি গ'ল নে নাই সেইকাৰণেতো মিঅ'চিছ সেইকাৰণেতো হা whole set to ছটা থাকিব কিন্তু but this cell is going to divide into first it will form two nuclei and then it will form two cells
but in case of mitosis anaphase of মানে chromosomal duplication
হৈছিলে সেই chromosome duplication হৈছিলে মানে কি what we have said is when the chromatids got separated right after that the chromatids were called as chromosomes আৰু সেই কাৰণে আমি chromosomes increase কৰিছে বুলি কৈছো কিন্তু meiosis is called as a reduction division so reduction of what reduction of number of chromosomes and the DNA content right okay rest of the students is this line understood Meiosis is also called as reduction division. Why is it called as reduction division? Because the chromosome numbers gets reduced. Parent cell or jiman ase, offspring or half ho jabo, no? So if you are talking about reduction division, you have to know at which stage the actual reduction takes place. It is anaphase. Prophase or hoy niki no hoy. Metaphase or no hoy. Actual reduction in the number of chromosomes will always take place in anaphase 1. Because we have talked about the bivalent separates and so the numbers reduced. While you are writing the notes, please compare the diagrams that you have drawn. Because against each phase, we are drawing the diagrams, right? So once you are done with this diagram, compare the diagram with the metaphase, the previous one. So you will know the difference, right? Metaphase or diagram akisilo na olo pakate. Anaphase or to kiba ta akim. So compare kori jabason, buzi paisa nai saba. Okay? Understood? Duita diagram, it looks almost the same, no? But there is a slight difference. Okay. And what is this fiber called as? এইবিলাক লিখিছোঁ আগতে Remember this word, interjournal fibers. Interjournal fibers আমি ক'ত দেখা পাইছিলোঁ, and what is interjournal fibers? Interjournal fibers ক'ত দেখা পাইছিলোঁ? Obviously in anaphase itself. Anaphase বুলি ক'লে we are talking about the anaphase of mitosis. What happens in mitosis? It equational division, right? That anaphase of কি হয়? Centromere splits. If the centromere splits, what will happen? It is a chromosome hoy, centromere is split kore, the chromatids would separate. Both the two chromatids remains linked with each other by certain fibers, those are called as key interjournal fibers. When they try to separate, the interjournal fibers becomes distinct, right? If you have forgotten, you can refer to the mitosis diagram that you have drawn, right? Ase to copy it, mitosis or junto anaphase diagram akisila swason. আছেনে? মেনশন আছে না ইন্টাৰজৰ্নেল ফাইবাৰ্ছৰ কথা? ইটছ দেয়াৰ ন? ইন্টাৰজৰ্নেল ফাইবাৰ্ছ কেতিয়া দেখা পাম? ফাৰ্ষ্ট, ইন্টাৰজৰ্নেল ফাইবাৰ্ছ is a characteristic of which phase? Generally, it is a characteristic of anaphase, right? Anaphase 1 or interjournal fibers dekha pam niki na pao? Kiyo kare na pao? Because the centromere does not, it's only the two chromosomes or the bivalent they got separated. It is not a single chromosome that got separated, right? Duta asile, hey duta hai separate hoise. Etar pra duta bhag hua nai, right? So this is another important point about anaphase 1. Okay, in my diagram, if you compare this size and this size, no, Kobe goise, that does not mean the size reduces. Okay, it's just my way of drawing a diagram. Size to ekei thakibo. Lage jodi dangoke aki diu aru ne buji paisu. The dotted line, what does it depict? The nuclear membrane, because we know telophase is just the opposite of prophase. Is this over? So telophase simultaneously ki hoy cytokinesis hoy, right, because telophase is the last stage of karyokinesis, right, together with telophase or karyokinesis or a process to hua loge loge cytokinesis will also take place. And how does cytokinesis take place in a cell? We have already discussed. If it's an animal cell, the process would be different, and if it's a plant cell, the process would be different. So after telophase simultaneous, not after, simultaneously cytokinesis hobo, tarpor duta cell form koribo, duta cell each nuclei, right. So this line is something that's very important. Did you understand this line? Telophase is the end of karyokinesis 1. Karyokinesis 1 would be followed by cytokinesis 1. After cytokinesis 1, what would be the result? Ki obtain koriba? Duta daughter cells, right? Hey daughter cells of so some point kita bujisun nai, right? We have talked about the parent cell. The parent cell got divided and formed two daughter cells. The two daughter cells, obviously tat ki hoise reduction in the chromosome number, and that's the reason why meiosis is called as a reduction division, right? The chromosome number reduced. Kintu, what about the DNA content? Double hoise nai double. Double kyo hua nai? Enike thaka nai niki? Right? Ultimately, etu separate hoi pelai edal hobo lagibo na lage. Right? So it is because you know what, okay, why are you getting confused? See, etia juntu cell paba, etu cellot chromosomes single haploid duta asile, eta ki ase haploid. DNA content ki ase double ase nai? Edal chromatid DNA content double hoy ase nai? Doubling of DNA by replication of DNA should occur at which stage? S phase. Etu ki phase? Etu division phase. Division phase ote jodi product tur already double hoy thake, that cell will again divide. Egita cell e akko divide koribo nokore? Koribo. So ultimately tar DNA content to besi hoy jabo na jai jodi. So ultimately after meiosis 1, we will obtain two daughter cells. The two daughter cells will have these two characteristics. Chromosome number would be reduced, but DNA content double hoy thakibo. Double kyo hoy ase karon the centromere did not split, right? If the centromere splits, then only half hoy jabo. Jabo na jai. Half hoy joa mane ki? If suppose we are talking about a chromosome like this and we are talking about a chromosome like this, what is the difference? This is also a chromosome, this is also a chromosome. What is the difference? About the amount of DNA, right? So DNA kar besi? Besi mane ki? Double nohoy jano, right? Etut ki? Single? This is double. So after cytokinesis 1, enukwa paisun nai chromosomes bilak, right? Ei bostu tu kenukwa hobo laibo? Enukwa hoi jabo laibo. So etu process baki ase. So meiosis completes itself in two phases. One is meiosis 1, the other is meiosis 2. Ekita buji palu? Okay. Now again the same question. Meiosis is called as a reduction division. Why is it called as a reduction division? Because the chromosome numbers reduces. Where does the chromosome number reduce, or in which phase do we see this? It's seen in meiosis 1. If in meiosis 1 itself the chromosome number reduces, then what is the importance of meiosis 2? Meiosis ok ami ki boli koiso reduction division. Ki okarne? Karon chromosome reduce hoy jay. Chromosome reduce kot hol? Meiosis 1 to hoye gol. Jedi hoy gol, meiosis 2 dorkar ki? What is the importance of meiosis 2? Why meiosis 2 needs to happen? Because of this line. Understood? Is it clear? Right? Why? What is the necessity of meiosis 2? Meiosis 1 to amak pura line to describe e kori dile no? Meiosis ok ami reduction division ko. Reduction to already hoye gol. Hole nai? Parent or jiman chromosome asile daughter cell ot half hoy gol nai? So reduce to hoye gol. So the definition is well explained, right? In spite of that, after reduction also meiosis 2 has to happen. What is the reason behind? Because DNA content to single koribo laibo. Double hoy ase no? Karon double thaka DNA content eta aru eta cellor jorokot jetia fuse hoy jabo, twice and 4n hoy jabo dokon. Right? These are all what? Germ cells, reproductive cells, right? Male gamete, female gamete. Male gamete jodi double content thake, he jodi female gamete logot fuse kore, male gamete having double content, female gamete having double content, both will fuse together, ki hoy jabo hon? tetra 4x condition হৈ যাব দেখোন বুজি পাইছানে right so ultimately to solve that problem we cannot be having a cell with 4n হলে তো species এ change হৈ যাব right so ultimately since gametes we have to talk about n no gametes are always haploid at this stage the cell is কি হৈ আছে diploid হৈ আছে না double n হৈ আছে so এই condition to কৰিব লাগিব কৰ পৰা twice n or পৰা এই বস্তুটো কোট হয় meiosis 2 হয় clear so next likhiba ha meiosis 2 please ask yeah please ask half the number of chromosomes mane ki jana ekdom simple ke just forget the notes remember the word bivalent what is a bivalent ba dutai na chromatids নহয় bivalent beleg chromatids are bivalent or logot eku relation nai no bivalent ki hoy bivalent is a pair right pair of what pair of chromosomes tamane dudal chromosome jedi ekelege thake tak ami ki boli kom pair word to use no kori pele ami ki use korisu bivalent hey pair to one chromosome is from the paternal side the other chromosome is for the maternal side I think you remember we have discussed this a pair of homologous chromosome one from the father one from the mother they will pair itself and ultimately they will form a bivalent yes or no right starting of the discussion no meiosis or junto cell opera start korise no meiocyte we have talked about at that time during prophase especially in case of zygote ami ki dekhisu chromosomes bilake pair korise right pair kori he pair to pairot keta thakibo duta right he pair to separate hoi pelai eta polot duta nuclei form korile nai right so if it forms two nuclei right and in both the nuclei from the bivalent eta he ase yat hoy no duita separate hoi gol so chromosome number reduce holle nai before this before the division no chromosomes number ki pair ke asile right pair ke thakute okay don't make it so complicated think about a human cell we are talking about a cell with twice n is equal to 46 human cell eta chromosomes kiman thake 46 right human cell is always diploid 46 hobo human cell to jete gamete produce koribo either we are talking about a male gamete or you are talking about a female gamete gamete is always haploid yes or no right haploid or diploid two sets of chromosome one set of chromosome hoy na noy definition right so we had a parent cell with two sets of chromosome now we have obtained a daughter cell with one set of chromosome that is what is called as reduction in chromosome বুজি পাইছা so aru ebar bhalke পঢ়ি সোৱা ha পঢ়ি চাই পেলাই not necessary you have to ask me the doubt right now itself right after the class also if you have a problem puri diba because you need to clear the concept karon jedi okman ghurai pokai question to ahe answer koribo nwariba নহলে right so meiosis ok ami ki boli koiso reduction division kior reduction koiso karon chromosomes number reduce kore chromosome number reduce kore mane parent cell is equal to 46 now the new cell that is formed tat kiman chromosome ase half 23 hoy na noy this is one cell originally a cell to kiman chromosomes asile 46 suppose if we are talking about the human cell right originally kiman asile 46 code asile eta nucleus or bhitorot yes or no yes okay hey nucleus to duta nucleale divide hoy gol equally distribute hoy goise okay and after this eto divide kori duta cell form koribo ei notun ke jun duta cell form korise a cell to aru a cell to chromosome number kiman hobo half of the parent cell okay so that's the reason why we have written this line clear divide hoi pelai cell form kora nai moi telophase diagram to he disu cytokinesis 1 ডায়াগ্ৰামটো দিয়া নাই ঠিক আছে যদি কনফিউজন হৈছে কৰি দিয়া ডিভাইড এইটো হ'ল টেলোফেজ 1 followed by কি সাইটোকাইনেসিস 1 সাইটোকাইনেসিস 1 ত আমি কি দেখা পাম দুটা সেল হয় দুটা সেলত এটা নিউক্লিয়াৰ মেমব্ৰেন থাকিব ইয়াতে এডাল থাকিব আৰু ইয়াতে এই ৰেড ডাল থাকিব এতিয়া ক্লিয়াৰ হৈছে নে ওকে clear so cytokinesis কি হল যে দুটা নিউক্লিয়াৰ উইথ ইয়াতে ডাবল ক্ৰোমোজোম আছে ইয়াতে সিঙ্গল হল so chromosome number got reduced right next what will happen the cell will undergo meiosis 2 কি হয় cell division কৰাৰ আগতে chromosomes বিলাক এনেকে থাকিব নে chromatin হৈ যাব chromatin হ'ব নে নহয় এতিয়া কি হৈ আছে chromosomes because we are seeing a proper diagram of the nuclear material right what we have written just see clear once the two cells are formed enukwa eta structure form korile after that what will happen decondense kora pisa again if you want to see the diagram here yar pisor diagram to kenukwa hobo some parts of red thread right in this case also what you will see clear so eto stage ok ami ki koisu chromosomes eto stage ok ami ki koisu chromatin so ultimately chromosomes transfer into ki hobo chromatin ta mane before the starting of meiosis 1 genetic material kini ami kuntu form ot dekha pam chromatin thread like because meiosis 2 also will have prophase yes or no the prophase ot ki hobo decondensation hobo nohoy yate ki hoise sorry yate decondensation hoise prophase 2 ki hobo condensation ei bostu to condense kori ako enikoi dekha pam ne na pam so it is the same thing that's getting repeated right so first we have seen this stage converted to this next after this ki hobo prophase 2 what will happen in prophase 2 a cell to keneke dekhapam right again you will see the proper chromosomes clear proper chromosome saba tar pisot ki hobo duita part divide hoy jabo right okay meiosis 2 likha okay which one this one or the lines okay from this point telophase 1 okay once you are done please say okay okay can I rub the diagrams okay বেছি কমপ্লেক্স হৈছে নেকি বস্তুটো বুজি পাইছা অলপ ঠিক আছে একো নাই আৰু একবাৰ পঢ়িবা হা পঢ়ি পেলাই আৰু বেছি ক্লিয়াৰ হ'ব আৰু ডাউটছখিনি লিখি থ'বা লিখি পেলাই মোক সুধিবা ঠিক আছে okay so before going to meiosis 2 see we have started from a cell right cellor nucleus asile nucleusot chromatin asile right etu ki hoy gol pairing hoy gol jiman genetic material asile prothomate chromosomes form korile chromosomes bilak pisat ki hol pairing hoy gol next ki hol দুটা নিউক্লিয়াৰ ফৰ্ম কৰিলে নেক্সট কি হ'ল ইউ হেভ ওৱান চেল উইথ ওৱান নিউক্লিয়াৰ ওৱান চেল উইথ ওৱান নিউক্লিয়াৰ obviously exchange already হৈ গৈছে I am not drawing it with two different colors I think it's understood right what next understood chromosomes কি হলে কনভার্ট হল chromatin ইয়াৰ পিছত কি হ'ব meiosis 2 hobo meiosis 2 first step ki prophase ot eta cell ki hobo nuclear membrane gusi jabo right bhangi to jabo nuclear membrane khon tar pisot jun bilak thread asile ako ami ki dekha pam chromosomes metaphase কি হব এখন পুরা গুছি যাব গুছি গৈ পে ইকুইটোরিয়াল প্ল্যানত অ্যারেঞ্জ হব রাইট আফটার মেটাফেজ 1 সরি মেটাফেজ 2 প্রোফেজ 2 কই আছো ন আমি ইয়াতে দেন কি হব এনাফেজ 2 কি হব যে মেটাফেজত কেনেকে আছিল এনেকে আছিল দুটা সো এনাফেজত কি হব separate ho jabo interzonal fibers bur dekha baba clear after anaphase 2 telophase 2 ki dekha pam duta cell chromosomes right ultimately duta cell to dekha na paw aru eta cell e dekha pam right duta nuclear ki hobo কারণ ক্যারিওকাইনেসিস ফার্স্ট হবো না তার পিছত কি হবো সাইটোকাইনেসিস 2 কারণে কি পাম দুটা সেল রাইট clear not clear we have started with this structure ultimately meiosis 1 2 সব হৈ যোৱাৰ পিছত আকৌ একেটাই বস্তু নিছাই পালো ন কিন্তু একেই জানো নহয় কিন্তু chromosomes or genetic material will always appear in the form of chromatin network itself prior to division only when prophase starts the chromatin network will condense and ultimately we can see the chromosomes chromosome word to ketia use koriba jetia ami chromatids dekha pam eta line of chromatids dekha paisa niki so we cannot be using the word chromosome here right ei bostu to hobo lagibo etia ha ami tak ki boli kom chromosomes right anaphase 2 ki hobo
anaphase 2 mane
chromosome সংখ্যা yeah chromosome সংখ্যা বাঢ়িব মানে actually we don't say about that from centromere point of view from chromatids point of view you can say that the chromosome number has increased exactly the same explanation in case of mitosis also right so jodi 4টা আছে 8টা হৈ যাব কিন্তু 8টা actually chromatid gitai he chromosome form korise notunke chromosome howa nai right you are drawing are you taking a screenshot of this one I mean are you taking a photo of this one this diagram and show it to your friends, and then they will have a very good opinion about me. তাহঁতক মেমে এনেকৈ পঢ়ায় গুড হা এইটো পঢ়াব লাগিলে হৈ গ'ল পেৰেণ্টছক দেখালে পেৰেণ্টছে তোমালোকক ইয়াৰ পৰা লৈ যাব আৰু ফ্ৰেণ্ডছেও ক'ব হ'ব ব'ল বহুত পঢ়িলা PDF-ত এইটো নাথাকে ন ভিডিঅ'টো আজি আছে কালি নাই ন অঁ সেইটোতো হয় অ'কে প্ৰচেছটো বুজি পাইছা অ'কে যদি প্ৰচেছটো বুজি পাইছা আই থিংক উই কেন মাইণ্ড আপ দ্য চেপ্টাৰ ইটছেল্ফ because meiosis 2 is exactly the same as mitosis. So if you have understood mitosis, what is the point of doing meiosis 2 again? নিজে নিজে পঢ়ি নালাগে লিখি দিম মই পুৰা ভালকৈ লিখিম অ'কে চলো মাইটোচিছ চৰি মিঅ'চিছ 2 হেডিং কৰা Meiosis 1 was also called as heterotypic.
Meiosis 1.
Meiosis 2 is homotypic. So you have to talk about the words. To see, 2C. Remember the word C, no? Yes. We are talking about the content of DNA. Is this part over? Okay, 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 last line to one, I know. Is the diagram understood? Prophase 2 যেটা start হবো cell to কেনেকে দেখা পাম something like this, yes or no? Is everyone clear about it? Right, because exchange has already taken place in meiosis 1. After that chromosomes got converted to chromatin. Chromatin will again get converted to chromosome, properly code dekha pam prophase 2, and rest of the explanation exactly the same. আমি যিখিনি ডিসকাস কৰিছো আৰু এইখিনি লাইনটো সব বুজিছো আৰু there is no point of describing understanding prophase again and again and again. From the first class itself আমি prophase discuss কৰি আছো. By now I think everything has been by hearted. Any point that you did not understand? Mitosis যদি বুজি পাইছো meiosis টো বুজি পাম except the last line baki rest of the lines understood in case of metaphase 2 the lines that you have written any point that you did not understand clear except last line বুজি পাইছা জানো spindle orientation is horizontal right why we have written this line because meiosis we have two metaphase now metaphase 1 metaphase 2 metaphase 1 or diagram to sason spindle to keneke form korisile vertically etu ki horizontally right so in case of spindle fiber formation or orientation ebar vertically form koribo ebar horizontally form koribo
What is the function of kinetochore?
Kinetochore, it is that area where the spindle fiber attaches and ultimately it helps the centromere or the chromosomes to separate. Kinetochore ki asile? Disc. Centromere or in and around disc shape structure. Yate ki lagibo? Spindle fibers bur lagibo. Right? Oh, spindle fibers I know. Understood? So ultimately what was the aim of meiosis 2? The aim of meiosis 2 was to reduce the content of DNA, right? So yate ki hol? Half a goal, so it got reduced. So ultimately the amount of DNA now is 1/4 of the original DNA. Mane ki we have started with a cell, etu jedi parent cell. A cell to jedi meiosis undergo koribo, first ot hi ki form koribo? Duta cell. এই দুটা সেলে আকৌ কি ফর্ম কৰিব, রাইট? সো ইয়াতে যদি এমাউন্ট অফ ডিএনএ যদি আমি 4C বুলি ধৰো, ইয়াতে কি আছিলে? ইয়াতে ইজ ইট ক্লিয়ার? ওকে, আফটার অ্যানাফেজ 2 লিখিবা টেলোফেজ 2। এখানে ওভার নাকি? Is the diagram over? ইয়াৰ পৰা question আহিব পাৰে nuclear membrane কোনে form কৰে right it is not always the মানে দীঘল points will study right সৰু points also will study right so who forms the nuclear membrane it's the endoplasmic reticulum that forms the nuclear membrane next because of meiosis what is the number of daughter cells that you will get tetrad okay. Because of mitosis so this is all about meiosis কিবা doubts আছে নেকি সোৱা meiosis to pura meiosis 1 and meiosis 2 just go through এবাৰ উপৰ সোৱা কিবা doubts আছে যদি খুধা is this part over diagram just go through the process and see whether it's clear or not. In the meantime, you have to write the significance of meiosis. Are the points clear? Significance understood? So almost the main contents we have completed. Cell cycle বুলি ক'লে, cell cycle and cell division we have talked about the cell cycle, the different phases of cell cycle and the M phase. Talking about the M phase, we have talked about the most important two, that is mitosis and meiosis, right? Apart from mitosis and meiosis, I think you all have heard another one that is called as amitosis. Shunisan na amitosis? Na nai shuna? Shunisa, amitosis is something that's seen in case of kunbilak organisms, bacteria like prokaryotic, right? And not specifically ami prokaryotes kobo nwaru karon exceptions to obviously ase. Amitosis or dutaman points likhibo lagibo. If this is over, heading points buli likhase. ER part of kiba questions ase niki? Mitosis, meiosis clear? Please go home, read and collect your doubts and bring it to the next class if you have any. Okay? ঘৰত পঢ়িলেহে এবাৰ ক্লিয়াৰ হ'ব ক্লাছত তো বুজি পোৱা নিচিনাই লাগে হয় না কোটো এটো লিখি আছা নে মই বুজাই দিব লাগিব কৈ দিব লাগিব ইন্ট্ৰোডাকশন অফ ভেৰিয়েশনস which is very important in evolution ভেৰিয়েশন আহিব নাহে exchange of genetic material is so sex sexual reproduction e ki ane variation helps in what evolution নেক্সট ক্লাছ কেইটা বজাত আছে 6:30 ইয়াৰ ৰিলেটেড এইটো চেপ্টাৰ ৰিলেটেড কেইটামান sums ahe sums in a sense problematic questions ahe thik ase so we have just half an hour nai so there are certain points you need to write and certain questions we need to discuss all questions kita jodi thoi diu we cannot afford another class to discuss the questions only because it's just 4 or 5 questions right as far as the points are concerned if I give you the PDF will you write it down right so please tate likhiye thwa write it down from the PDF or else you'll forget I know core PDF code jabo eku thik nai kya hua kitna page hoga लंबा है तो 1 पेज फुल बस okay in the point section what you are going to write is some points about mitosis not very important at all so you don't have to study also khali eta line lagibo mitosis is also called as direct cell division right mitosis or hetu point important what happens in mitosis I think you remember that binary fission type no right nucleus to ki hobo spindle form koribo constrict koribo dubhag hobo ako ta cytokinesis hobo okay occurs ekini likha ase ekini tumalke nije poriba that's the first point second point to dia ase types of meiosis or under certain words like gametic meiosis zygotic meiosis remember the two words haplontic life cycle and diplontic life cycle we have discussed that so ekini portion we have already discussed we have talked about 3 types of life cycle no haplontic diplontic and diplohaplontic or haplodiplontic accordingly they had 3 different names also gametic zygotic aru sporic right so hekini information ami plant kingdom already korisu so that is another point that's written here just write it down read and see if you understood or not because explanation we have done in the plant kingdom third point is about meiosis is commonly studied in hetu point important onion buds thik ase PDF tur pora likhi loba ha that is important point rest okay thik ase so tinta point direct cell division mitosis the different types of meiosis that is zygotic sporic aru gametic and the third point is it is studied in onion cells bas etia ketaman formula likha rakhi diba ha jega please likhi loba another point is in your NCERT book the diagrams are given no cell division diagrams kita ase so these diagrams that I have drawn is from the point of explanation and understanding right NCERT junkhon diagram dia ase hetu he saba porikhale right junkhon chart dia ase no it's hebilak no which page okay because if diagrammatic based question ahe it is from the NCERT that the direct diagram would be taken right so please refer to the diagrams from the NCERT book as far as explanation and understanding is concerned you can refer to my diagrams right? eto likhasun joldi প্ৰথমতে লিখি লোৱা যোনটো ফৰ্মুলা দিছে ফৰ্মুলাটো এক্সপ্লেইন কৰিছোঁ বুজি পাইছা নাই চোৱা ৰাইট যদি কুৱেশ্যন আহে 200 চেলছ ফৰ্ম কৰিবলৈ কিমান মাইটোটিক ডিভিজনছ লাগিব ইট ইজ n-1 ক্লিয়াৰ মই কি লিখিলোঁ সৰি ইয়াতে নাম্বাৰ অফ মাইটোটিক ডিভিজন সুধিছে ইয়াতে নাম্বাৰ অফ জেনেৰেশ্যন সুধিছে ৰাইট ডিফাৰেঞ্চ জানো ন ডিভিজন এণ্ড জেনেৰেশ্যন মিনছ উই আৰ টকিং এবাউট দ্য অফস্প্ৰিং ন 1ৰ পৰা কেইটা ফৰ্ম কৰিব দুটাৰ পৰা 4টা 4টাৰ পৰা 8টা চ' দিজ আৰ দ্য জেনেৰেশ্যনছ Meiosis related যদি question আহে what is the number of seeds that are formed what is the number of fruits that are formed what is the number of grains that are formed MCQ যদি কৰিছা এটো question আহে ঠিক আছে from need point of view ইমান frequently asked question নহয় কিন্তু আহিবও পাৰে right কিন্তু books বিলাকত বহুত আছে এটো question right so if that is asked what you need to remember this one whenever we talk about meiosis অকল এটো বস্তু ৰাখিবা what does this tell us এতিয়া আমি যেতিয়া examples কৰিম it will become more clear but this tells us about what the number of seed form koribo libo seed is a result of ki fertilization koribo lagile gametes lagibo male gamete and female gamete something related to microsporogenesis and megasporogenesis which you have not studied so male gamete and female gamete if you are talking about that eta meiocyte male gamete keta form koribo 4 cells right in case of male gametes ki hoy all the 4 remains functional in case of female gamete out of the 4 that are formed from one meiocyte only 1 remains functional and 3 of them degenerate right so eta loisu eta kihor karan loisu karan tinta degenerate hoy gol aru eta kihor pra loisu je all the 4 remains functional moi question keta man dim tetia easy hoy jabo ঠিক আছে ekini hol okay write a question now Actually, junto degeneration of kotha koise no, this is not a part of first year. Etu actually informations kini details or second year course othe ase. Junto first lesson sexual reproduction boli eta chapter ase. Hey chapter tu he etu bostu discuss kora ase. ঠিক আছে আৰু এটা কথা কি these questions no or these things that we are writing especially this question is also related to sexual reproduction so cell division ot nahibo pare kintu sexual reproduction পঢ়াৰ পিছত এটো question আহিব pare because sexual reproduction also will be studying about formation of gametes itself male gamete keneke form kore, female gamete. I think we have already done that, right? So jehetu cell division logo related, we have written it down. Kintu more precisely, this question is related to the second year course, sexual reproduction of kind. Okay, just to have a brief idea, ki janibo laibo swa. There is a parent cell. The parent cell will undergo meiosis. Parent cell ki hoy twice n. If that cell will undergo meiosis, that cell would be called as what? Myocyte, right? If you are talking about male reproduction, then obviously that cell will produce the male gametes, right? A myocyte will undergo meiosis and produce how many male gametes? 4 male gametes, right? Similarly, in case of female also, a myocyte will undergo meiosis and produce how many female gametes? 4, right? But what we have observed that out of the 4 female gametes that are formed, it is only one that survives aru baki 3টা degenerate kori jay. Kintu male gametes gutei 4টা jungita form kore gutei kita functional hoy thake. This is the simple line that as of now you need to understand. Details in that chapter, right? এটা তো এক্সপ্লেনেশন নালাগে বুঝি আছো First question ki asile number of cells that are formed. Etu to ami janu n-1, right? Yate ki khudise generations of ki asil to the power of n, right? Yate kiman lagibo 128 cells form koribole ki either you can use the formula or you can go by this method also, right? Eneke koileu to ami pam keta generation hobo 128 double kori thaka 4 opera 8, 8 opera 16, 16 opera 32, 64, 128, hoy na hoy? Correct? So generation keta? 7? Generally 127 by 8, that is the farthest, tatke beshi aru nidiye, thik ase. So jodi by chance formula to pahoriu jua, whenever you hear this word generation, this is the way you can just calculate, right? So etu kori maximum 128 or pisor to dibo 300 something kiman hobo 128 into 256 ha. So 7 by 8 generation or lake dibo. পুৰা লিখি দিম ফাৰ্ষ্টটোত যাতে নেকি বুজি পোৱা ইমান লিখি থাকিব নালাগে কিন্তু একদম চাই ল বুজিছা নাই after class लिखा कि नहीं ठीक है One okay সেইটোৱে মই কৈছোঁ না এইটো একচুৱেলি বস্তুটো ছেকেণ্ড ইয়েৰৰ পোৰ্চন দেই মাইক্ৰস্প'ৰ মাদাৰ চেলে মাইক্ৰস্প'ৰ ফৰ্ম কৰে মাইক্ৰস্প'ৰৰ আৰু এটা নাম কি পলেন গ্ৰেইনছ ৰাইট মেগাস্প'ৰ মাদাৰ চেলে কি ফৰ্ম কৰে মেগাস্প'ৰছ ফৰ্ম কৰে আল্টিমেটলি উই আৰ টকিং এবাউট দ্য এগ ৰাইট চ' প্ৰচেছটো যিহেতু ডিটেইলছ ক'ত দিয়া আছে ছেকেণ্ড ইয়েৰ কোৰ্চত চ' উইল বি ষ্টাডিং এবাউট ইট এজ অফ নাও জাষ্ট ৰিমেম্বাৰ দিছ এটা লাইন মনত ৰাখিলে হ'ব এটা মিঅ'চিছ ইফ 1 mmc ইন কেছ অফ মেল গ্যামেট 1 mmc উইথ ৰিজাল্ট ইনটু 4 মেল গ্যামেট In case of female, 1 mmc results into 4 but remains functional keta. Eta. So ultimately, I think we can conclude that if you are talking about the female part, tetia hale eta mmc e keta megaspore form koribo. Eta. Ultimately keta form kore 4 ta kintu 3 ta degenerate kori jay na jay. To eta hole nai. So ami etu kobo paru ne noaru 1 mmc will produce 1 megaspore by the process of meiosis. There are two words, one is microspore mother cell and the other is megaspore mother cell, right? Microspore mother cell ki form koribo microspores form koribo microspores ki hoy pollen grains megaspore mother cell ki form koribo megaspores form koribo megaspores ki hoy nothing but egg ekdom simple ke right so right egg actually egg নহয় megaspore to ভিতৰত egg থাকিব হেবোৰ পিছত পঢ়িম Sexual reproduction in case of plants. If we talk about sexual reproduction, we have to talk about the flower, we have to talk about the male part and the female part, right? Male part boli kole stamen, or female part boli kole carpel. Stamen or parts ki? Right? Anther, filament, anther of bhitorot ki thake? Pollen grains. So ইয়াতে কিবা কিবি process হ'ব লাগিব যাৰ কাৰণে pollen grains বিলাক form কৰিছে. Pollen grains কোনে form কৰে? কিছুমান cells এ form কৰে. সেই cells বিলাকক কি বুলি কওঁ? Spore mother cell or microspore mother cell, right? And microspore mother cell or ploidy কিমান হ'ব? Twice n. তাহাতে কি হ'ব? Meiosis কি form কৰিব? N. ঠিক তেনেকে if you are talking about the female part, we refer it to as carpel, right? Carpel ডাল কেনেকুৱা আছিলে? Stigma, style and ovary. Inside the ovary ki thakibo? Ovule. Ovule bhitorot ki thakibo? Egg. So ultimately a structure to ba a egg to kunubai form koribo lagibo. Kune form koribo? Megaspore mother cell. Megaspore mother cell or ploidy kiman? Twice n. Twice n e undergo ki koribo? Meiosis. Eneo eta cell meiosis undergo korile keita cell form kore? Sarita. So in this case also they will produce 4 cells. In that case also they will produce 4 cells. But in case of male, all the 4 cells are functional. In case of female, 3 of them degenerate, only 1 remains functional. So eneke kobo parone nwaro. In case of female reproduction, 1 meiosis produce 1 egg. Yes or no? In case of male, 1 meiosis produces how many pollen grains? 4. In case of female, 1 meiosis produces how many egg? 1 actually to ami jano 4 ta form kore kintu 3 ta to degenerate kore no. So that is the reason why we are writing here. So since we are talking about 50 eggs, no, 50 eggs kiman meiosis lagibo? 50. Ar jodi ami tomar 50 pollen grains koisu তেতিয়া হলে এটাত তো 4 টাকে প্রডিউস করে ন, তো 50/4 হবো নহয়? Is the point clear? Right? এখানে সেকেন্ড ইয়ারের পোর্শন। Okay, এইটো কোয়েশ্চন কৰাচোন। এবাৰ কি দিছো? Okay, questions বিলাকত seeds বুলি নাথাকি grains বুলিও থাকিব পাৰে, fruits বুলিও থাকিব পাৰে। In each case, the answer would be the same because seed is a product of fertilization. Fruit is also a product of fertilization. ইয়াতে কি হ'ব? n plus n/4? হয়? এইখিনি পোর্শন অলসো ফর সেকেন্ড ইয়ার পর লিখি তো লাগিব এইখিনি কিন্তু অফিসত হা এইখিনি বুঝি পাইছো Okay, from the point or up till the point you have understood what we have discussed. If you are talking about microspore mother cell, it will form one meiosis produces 4 pollen grains, all the pollen grains are functional. If you are talking about megaspore mother cell, meiosis produces 4 megaspores but only one remains functional. Pollen grains are ki hoy? 4ta. So, what is the number of meiosis required to form the pollen grains? N by 4. Clear? It is question? Yate ki hobo lage? From the up till the point that we have understood, divided by 4 hobo lage ne? Kintu yate it was to an exception. He karne a question to ahe. Why is it exception? Because in this plant only, right? Only in case of cyperus, it is out of the 4 pollen grains, only 1 pollen grain remain functional. He karne question to okol cyperus jodi likhi ni dile heten, tetiale n by 4, right? Very good. Right? Enukwa dhৰণৰ অংক বিলাকত you don't have to tackle, I mean rack your brains, okay? What you need to remember, এটা container আছে, ঠিক আছে? Containerটোত কি দিয়া আছে? Suppose bacteria full কিমানত হয়? 60 minutes of half কিমানত হ'ব? 60 minus 1, right? 1/4 কিমানত হ'ব? 60 minus 2. 1/4 প্ৰথমতে এটা আছিলে, এটাই ডিভাইড কৰি 1/4 ফৰ্ম কৰিলে, 1/4 পিছত হাফ হ'ল, হাফৰ পিছত ফুল নহ'ল জানো, তিনিটা নহয় জানো? চ' ফুলটোত কি আছিলে? 60 মিনিটছ, 60 মিনিটছৰ পৰা 1 মিনিট কমাই দিলে হাফ হ'ব, 1 মিনিট 60 মিনিটছৰ ইয়াৰ পৰা 1 মিনিট কমাই দিলে 1/4 হ'ব, ৰাইট? ইফ ইউ আৰ টকিং এবাউট দিছ, ফুলৰ পৰা যদি কমিবলৈ লাগে, দেন হোৱাট ইউ ডু ইজ কিপ অন মাইনাছ, ৰাইট? মাইনাছ কি কৰিব? 1 1 মাইনাছ কৰি থাকিবা। যদি উল্টাকৈ আহে কুৱেশ্যনটো যে 1/4 ফৰ্ম কৰিছে, ফুল ফৰ্ম কৰিবলৈ কি লাগিব? +1 1 কৰি দিবা। ইজ এ পইণ্ট আণ্ডাৰষ্টুড? আৰু ইয়াতে যদি ইয়াতে হাফ কাপৰ কথা সুধিছে ন, 1/4টো সুধিব পাৰিলে হয় কি হ'ব? 60 minus 2 or 59 minus 1. Just remember, either you will keep on plus minus 1 or you will keep on minusing 1 1, right? Again, অকমান যদি complex কে question to দি দিয়ে, right? You have to see this word also. Sometimes question কি আহে, a bacteria divide twice in every minute. তেতিয়াহ'লে -2 হ'ব। Sorry, not this one. in every 2 minutes. If the question is asked like that, no, a bacteria divide once in every 2 minutes. Tetla minus kiman koriba 2, plus kiman koriba 2. If the question to dia ase a bacteria divide once every minute, minus kiman koriba 1, plus kiman koriba 1. Right? Maximum case of okol a question to ahe, right? 2 minutes or generally questions nahe. Is the point understood? So since we are talking about cell division, so we need to talk about bacterial division also. So bacterial division or a question nahe aru beleg eku nahe. Because our furthermost generation of question ahibo pare, generation of question kenege kore to dekhai dilu. Right? Just multiply kori jaba, double kori jaba, 1 to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32, 32 to 64, 1 to 88, 256, 364, etc., etc. Right? So I think that's all for today also. And okay, once you get the PDF, aji PDF pai jaba, PDF for points khini likhiba, and you have some more sums, right? Exactly the same pattern, aru ketaman onko ase, sai loba buji paisa ne dai. Right? So this brings an end to this chapter. Next class, next chapter, ki ase course of transport in plants heavy ase ne, term to ase nai, term to ki ase first, photosynthesis. So let's start with photosynthesis. Okay, so next class we'll start with photosynthesis. Thank you. Mineral nutrition, transport in plants, hegita nai no. Okay.